Hey everybody, it's Mr. Lane here. We're talking about some DNA structure. I got really excited today, so excited to talk about DNA that I wore my jeans today. Get it? Because genes, DNA, it's in your genes. We're going to talk about DNA. Here we go. DNA, it consists of two molecules that are arranged into a ladder-like structure that we call a double helix. So when we say DNA, it's a common misnomer that's only one molecule. It's actually two molecules that are stuck together. A molecule of DNA is made out of building blocks. We remember this from chapter 2 when we talked about chemistry of life. Those building blocks are called nucleotides. We've already met one of these nucleotides when we talked about cellular respiration, when we were talking about ATP. So the adenosine was a nucleotide that our body uses for energy. So each nucleotide contains three different subgroups. There is the phosphate group, the pentose sugar, and the nitrogenous base. This simple diagram right here is what I would expect you to know for the building blocks of a nucleotide, or the building blocks of DNA. You have the phosphate group here, the pentose sugar, and the nitrogenous base. This should be kind of review from cellular respiration. Remember, ATP simply had three phosphates. So this would be like AMP, or adenosine monophosphate. A nucleotide is the phosphate and the sugar form the backbone of DNA. So the backbone of DNA is literally the bases of a ladder. The rungs are where the nitrogenous bases form. So the phosphate and sugar make up the backbone of the ladder. The nitrogenous bases make up the rungs of the ladder. Okay? So there are four flavors of nitrogenous bases that we are going to talk about. The first one is adenine. We've seen this one before with adenosine triphosphate. So adenine is like adenosine triphosphate with missing two phosphates. So adenine. We also have cytosine, okay, or sometimes just called C. So A we abbreviate with A, cytosine we abbreviate with C. Next one is thymine. And finally, we have guanine. One thing that I want you to pay attention here is A will always bond with T, and C will always bond with G. What we call that is a base pair. So the fact that A only bonds with T and C only bonds with G will make it very, very good for us to be able to decode DNA later in the semester. Hydrogen bonds are what are keeping these nitrogenous bases together. So here is the chemical structure of thymine and adenine together. Here is the adenine, here is the thymine. You'll notice that this oxygen, if we remember all the way back to our water lab, that oxygen likes to keep its electrons a little bit closer to itself. That creates a slightly negative charge. And hydrogen here is going to have a slightly positive charge. So this weak attraction right here, that's what's keeping those two molecules together, keeping the ladder together. And in this case, between cytosine and guanine, we have 1, 2, 3 hydrogen bonds, whereas with thymine and adenine, we only have 2 hydrogen bonds. So remember, these hydrogen bonds are weak, much weaker than the covalent bonds that are going to hold up the backbone of DNA. DNA structure, we call this complementary. We call it complementary because we know every time we see an A, we know that on the opposite side is going to be a T. Every time we see a G, we're going to have a C. Every time we have a T, we're going to have an A, and every time we have a G, we're going to have a C. So then what we can do is if we know one half of the DNA, we automatically know the other half. So let's give this a try here. We have T, G, A, G, T, C, A. We know then that A has to be binding to that T, C with the G, T with the A, C with the G, A with the T, G with the T, C, T with the A. Now, something interesting here. We're like, well, what the heck, Mr. Lane? That side right there is upside down. That's a very true statement. And it's true because this is the way that the molecule naturally fits together. They're not going the same way. They are what we call anti-parallel. And we're going to talk about this. You'll see this in several models in class. So to crack the genetic code that we have in our DNA, we need to look at the sequence of the base pairs. The information for life is shared in those and stored in those letters in that DNA sequence. And this is how we know different life is all related, because if we look at the DNA of a human and the DNA of a chimp, we share more base pairs in common than we do with any other organism. Whereas if you look at us and say an oak tree, we still share some things, but just not nearly as much. There are some things that are so well conserved throughout all living things that if you look at the DNA sequence, it's changed very little in billions of years. So the bases are arranged into triplets, which we call codons. Just like if I gave you a book and all the letters were all just jumbled together without being separated into words, it'd be very hard to read. So the way we read DNA is in words. We call these words codons, and they are sets of three nucleotides. So in this case, it would be AGG, CTC, AAG. We read it like that in those sets of three. Now, my question for you is, what is the sequence that would go with this? What is the complementary sequence? Take a minute, answer the question, okay? Here's the answer. TCC, GAG, TTC, AGG, ATC. If you didn't answer that right, make sure you go back and check and know what those base pairs are. So DNA structure. DNA is the whole molecule. In fact, when we say the whole molecule, we always have this misconception that DNA, we only have one molecule of DNA in our bodies. In fact, when we say we have 46 chromosomes, what we're really saying is we have 46 different strands of DNA, okay? DNA's primary function is to create proteins. It's storing the information to make proteins. And a gene is simply one stretch of that DNA. So a small segment of that DNA, it codes for one protein. So each unique gene has its own sequence of bases, or what we are going to call a genotype, okay? So let's say that we have a gene that says make hair. Well, I have the genotype that says make brown hair, where some of you might have the gene that makes black hair, or blonde hair, or red hair. So you have the genotype. We all have the same hair gene. It's in the same spot on the chromosome, but we have different information in that gene that leads to these different genotypes, which ultimately leads to what we call a phenotype. Okay, so the unique sequences of bases will code for the production of a unique protein. That unique protein has different shapes. Those different shapes have different functions because remember, structure equals function. So the genotype is the letters that I have in my gene, in my DNA. The phenotype is what that relates in. So what the most easy way is like things like hair color or, skin color or eye color, or you can do this weird thing with your thumb. Those are all phenotypes, but those are led and created by genotypes. Other cases, it's much more subtle. A phenotype could be simply the structure of a protein. Cystic fibrosis leads to a lot of complications with your lungs, and that is due to a phenotype that is only at the molecular level. You don't actually see somebody necessarily with the phenotype, but it's due to one molecule being off just a little bit because there's something wrong with their genes. So here's the levels of organization that you have to know for this unit. You need to know what chromatin is. Chromatin is tightly wound DNA. It's DNA wrapped around these really cool histone proteins. It actually looks like a spool. So here's the DNA right here, wraps around those histone proteins. It condenses, and then that chromatin then condenses into chromatin fibers. So these nucleosomes condense into chromatin. So DNA gets wrapped around those histone proteins, which condenses into chromatin. So there's some words that you need to know. Here's another example of the exact same thing. It's all found in the nucleus here. This is a chromosome. This is actually a duplicated chromosome. We're going to come back and talk about why we know it's a duplicated chromosome. We have the DNA getting wound around histone proteins, which condenses into chromatin, which condenses into a chromosome. Chromosomes. Chromosomes are super coiled. Remember, think about this. If you had 23 volumes of a book, or 46 volumes of a book rather, you had 46 different books. You would not want to have all the pages just willy-nilly flying all over the place. You'd want to make everything pretty tight and compact. DNA does the same thing. There's a lot of information stored there, and so what it's doing is we are storing all that information highly wound. So only the pieces that we're using, we are opening the books and only reading certain pages at a time, rather than flipping through all the pages all the time. Prokaryotes, or those bacteria and archaea that we learned about, they have no nucleus. What they have is a very simple circular chromosome. So it makes a circle. Whereas eukaryotes, we carry our chromosomes in the nucleus, and we have linear chromosomes, where they are in a straight line. And there's some very big drawbacks for having a long linear chromosome that we are going to talk about. So here's the big picture. This is where we're going for like the rest of this semester. This is kind of the foundation of where we're going. We're starting with DNA. Small chunk of DNA, we call that a gene. That gene codes for a protein. That protein, or the that genotype that leads to that protein will lead to a trait, or what we call a phenotype. And if you don't remember how to make a phenotype, you always remember this. Phenotype is the physical characteristic, or as I like to think about it, you want to get physical phenotype. You want to get physical phenotype. Yeah, I'll stop there because I'll really get into my phenotype dance. So your goal for the rest of the semester here is explain how do we get from a genotype to a phenotype. That's right. You want to know, how does this cell, how does the information in my nucleus lead to me having a certain eye color, having a certain height, having a certain hair color, having curly hair, straight hair? How does that happen? We're going to spend the most of the semester checking it out. So this is where we're going. This learning how DNA works is just the very beginning. I hope you enjoyed this very brief set of notes. And here is your moment of monkey. Have a great day, guys. We'll see you soon.